Close your eyes, feel the weight of your body settle. Let the present moment fade away. Now, step backward through time. Not years, not centuries, but thousands of years. Keep going, further, deeper into the past. Millions of years dissolve like mist. You are standing somewhere you have never been, somewhere no modern human has stood in living memory. Open your eyes. You are on the edge of something ancient beyond all comprehension. The water in front of you is warm, not pleasantly warm, warm like a fever. The surface shimmers under a sun that burns 30% brighter than you have ever known. The sky above is not quite the blue you recognize. It holds a faint, hazy amber tinge, a signature of an atmosphere still finding its balance. And below you, stretching down into unfathomable darkness, is an ocean that predates every creature you have ever seen, heard, or imagined. You are standing at the edge of the Iapetus Ocean. It is 480 million years ago. The Ordovician period is in full tide. The ocean before you is ancient even by the standards of ancient things. It is roughly 5,000 kilometers wide at its broadest point. It separates land masses that will not collide for another 80 million years, and it is alive. Extraordinarily, bewilderingly, magnificently alive. The water moves. Something rises from the deep, slowly, deliberately, a shape that registers in your instincts before your mind catches up. It is long, perhaps 6 meters from end to end. It moves with the fluid economy of a creature that has perfected itself over millions of years. This is Orthoceras, a straight-shelled nautiloid cephalopod, a predator of the ancient sea, and it is hunting. Its shell gleams faintly in the filtered light. Its tentacles trail behind it like silk in a current. It does not know you are here. It has never needed to know anything like you. Watch it descend. It sinks back into the dark water below 200 meters and is gone. Just like that, swallowed by a depth that holds more secrets than any place on the surface of this ancient world. The numbers alone are staggering. At the height of the Ordovician, sea levels were approximately 170 meters higher than they are today. Not slightly higher, not incrementally higher, 170 meters. If you could stand on what will one day become the city of London, you would be submerged beneath several dozen meters of warm, oxygen-rich, creature-filled ocean. Everything you think of as dry land, the continents, the coastlines, the familiar shapes of the world, does not exist here, or rather, it exists in a form so radically different that it would not register as the same planet. 85% of all land on Earth at this moment is covered by shallow tropical seas. That number deserves to sit with you for a moment. 85%. The continents are compressed into unfamiliar clusters. Gondwana, an enormous supercontinent containing what will one day become Africa, South America, Antarctica, Australia, and India, sits partially over the southern pole, ringed by ocean. Laurentia, the ancient core of what will become North America, drifts as a separate island continent near the equator, bathed in shallow warm water on every side. Between them and around them, the ocean reigns, and that ocean is teeming. The Ordovician seas represent one of the greatest explosions of marine diversity in Earth's entire history. In these waters, life has found forms that no modern mind fully anticipated before the fossils began to speak. There are filter-feeding graptolites, colonial organisms that string themselves along the current like strange living garlands. There are trilobites, hundreds of species of them, crawling across the seafloor in every size from a fingernail to a dinner plate. There are sea scorpions, eurypterids, that will one day grow to over 2.5 meters in length and will remain the largest arthropods this planet has ever produced. There are echinoderms anchored to the seafloor like stone flowers, opening their arms to the passing current. There are brachiopods clicking softly in their billions, so numerous that their shells will eventually form the limestone bedrock of entire mountain ranges. This is not a simple world. This is not an empty world. This is a world of extraordinary complexity, and you are standing at its edge, watching its surface breathe. Now look down. Below you, in the water column, something is changing. The further down you go, the stranger it gets. At 300 meters, the light fades to a cold, blue-gray twilight. At 600 meters, there is no light at all. At 1,000 meters, the pressure is nearly 100 times what you feel on the surface. At 2,000 meters, the temperature drops to just above freezing, and the creatures that live there bear no resemblance to anything near the surface. They have evolved for a different world, a world of eternal darkness, crushing pressure, and food that drifts down from above like slow snow. This vertical gradient, this layered strangeness, has existed in Earth's oceans for more than 500 million years. And here is what makes it truly extraordinary. The deeper you go in a prehistoric ocean, the more alien the world becomes. Not just in the modern sense, where we speak of deep-sea creatures as strange and unusual, but in a fundamental evolutionary geological sense. The deep ocean is a refuge, a time capsule, a place where ancient forms of life survive long after their surface cousins have vanished, changed, or been wiped out entirely by one of the 5 mass extinction events that will reshape this planet's surface again and again across geological time. The deep sea does not forget. The surface of the ocean changes with every major catastrophe, with every asteroid impact, every volcanic eruption, every sudden climate shift. But the deep absorbs, the deep persists, the deep holds things. Which brings us to the question at the heart of this journey. If the prehistoric ocean was already extraordinary at the surface, teeming with life forms we can barely imagine, shaped by forces no modern human has ever experienced, what on earth was living in its depths? And more importantly, why does the story of those depths grow stranger, more complex, more impossible to comprehend with every additional meter you descend? That is what we are here to explore together tonight. If you enjoy journeys like this one, consider subscribing. There is much more waiting for you. And to understand why prehistoric oceans grow stranger with depth, we must first understand what science has told us about the ocean's structure, both today and across the vast sweep of geological time. The study of ancient oceans is a field called Paleoceanography, and it is one of the most demanding and fascinating disciplines in all of Earth science. Paleoceanographers do not simply dig up bones or collect fossils from riverbanks. They drill into the sea floor itself, extracting cylindrical cores of sediment that record, layer by compressed layer, the chemical and biological history of the ocean over millions of years. Each centimeter of core can represent thousands of years of oceanic history. Each layer contains chemical signatures, microscopic fossils called foraminifera, and isotopic ratios that tell a trained scientist the temperature, salinity, oxygen levels, and circulation patterns of ancient seas with extraordinary precision. The field was transformed in 1968 when the Deep Sea Drilling Project later expanded into the Ocean Drilling Program, and eventually the Integrated Ocean Drilling Program began systematically extracting sediment cores from ocean basins around the world. Over the following decades, thousands of cores were brought to the surface. Entire chapters of Earth's oceanic history were revealed for the first time. The work of scientists like Nicholas Shackleton, whose landmark analysis of oxygen isotope ratios in foraminifera in the 1970s allowed researchers to reconstruct global temperature records going back millions of years, fundamentally changed our understanding of how the ocean behaves across geological time. The findings were not merely interesting, they were revolutionary. Among the most important discoveries was that the deep ocean, the vast abyssal zone below 2,000 meters, is not a stable, unchanging environment. It is profoundly sensitive to events at the surface, but it responds slowly, immensely slowly. Changes that happen in centuries at the surface can take tens of thousands of years to propagate into the deepest basins, meaning the deep ocean carries a temporal memory, holding chemical, thermal, and biological signatures from events the surface has long since erased and forgotten. This principle becomes even more extraordinary when applied to prehistoric time. Consider the Cretaceous period, a span of approximately 79 million years, stretching from roughly 145 million years ago to 66 million years ago. During the Cretaceous, global average temperatures were approximately 8 to 10 degrees Celsius warmer than today. There were no permanent ice caps at the poles. Sea levels were approximately 200 meters higher than at present. The continents were arranged differently than today, and the ocean circulation patterns that govern the global distribution of heat, nutrients, and oxygen were fundamentally unlike anything the modern world experiences. In these warm Cretaceous seas, a phenomenon known as oceanic anoxic events, or OAS, occurred repeatedly. During an OAE, large portions of the ocean, particularly in the deep basins, became severely depleted of oxygen. The evidence for this is preserved in the sediment record as dark, carbon-rich layers called black shales. The largest Cretaceous OAE, known as OAE2 or the Bonarelli event, occurred approximately 94 million years ago and lasted for roughly 800,000 years. During this event, the oxygen minimum zone in the ocean expanded dramatically. Creatures living in the deep water column were suffocated. Mass die-offs of marine organisms occurred across multiple depth levels, and yet life persisted. In some zones, it thrived. Anaerobic bacteria, organisms that do not merely tolerate the absence of oxygen but require it, expanded their range into waters that had previously been hospitable to more complex life. The science of this event owes much to geochemist Elisabetta Erba at the University of Milan, whose analysis of calcareous nanofossils helped reconstruct the Cretaceous Ocean's changes with remarkable precision. Her work showed that the Cretaceous Ocean was not simply a warmer version of the modern ocean. It was a fundamentally different system, operating under different physical and chemical rules. Going further back in time, to the Cambrian period, roughly 541 to 485 million years ago, we encounter an ocean that was almost unrecognizably different from the one that exists today. The Cambrian Ocean contained no fish, no vertebrates of any kind, no plants in the modern sense. Its chemistry was unusual by modern standards: relatively low oxygen at depth, high concentrations of dissolved minerals, and a complex interplay of sulfur cycling that modern microbiologists are only beginning to understand. And yet, it was in these waters that the blueprint for nearly every major animal body plan on Earth was first assembled. The discovery of the Burgess Shale in 1909 by Charles Doolittle Walcott, a geologist working for the Smithsonian Institution, high in the mountains of what is now British Columbia, Canada, gave science its first detailed window into this world. The shale preserved, with extraordinary fidelity, the soft-bodied organisms of a Cambrian seafloor community from approximately 508 million years ago. Organisms like Anomalocaris, a predatory arthropod, reaching lengths of up to 1 meter, with grasping frontal appendages and compound eyes. And Hallucigenia, a creature so baffling to early paleontologists that its discoverers initially reconstructed it upside down, mistaking its spines for legs and its legs for tentacles along its back. Walcott's find was significant. But the deeper interpretation of what the Burgess Shale represented, a window into the Cambrian explosion, the most rapid diversification of animal life in Earth's history, was developed through the extraordinary work of Stephen Jay Gould, Harry Whittington, Simon Conway Morris, and Desmond Collins across the second half of the 20th century. Their work revealed that the Cambrian Ocean was not simply an early version of the modern ocean. It was a testing ground, a laboratory, a place where evolution was running its most radical experiments simultaneously. And the strangest experiments were happening in the deepest water. Modern research has confirmed that deep-sea environments in the Cambrian were among the most chemically extreme on the planet. A 2019 study published in the journal Nature Geoscience, led by Dr. Noah Planavsky at Yale University, used chromium isotope ratios in ancient marine sediments to reconstruct oxygen levels in Cambrian deep-water environments. The findings showed that oxygen concentrations in the Cambrian deep ocean were as low as 1 to 2% of modern atmospheric levels. Not low, vanishingly low. A level at which most modern complex animals would suffocate within minutes. And yet, complex animals lived there, or something remarkably close to complex animals. The study of ancient deep-sea life is made difficult by preservation. Deep-sea sediments rarely protect soft-bodied organisms, and the pressure, chemistry, and slow sediment accumulation tend to destroy biological material before fossilization. Our knowledge of ancient deep-sea communities is therefore far less complete than our knowledge of shallow water life. The deeper fossil record is fragmentary, partial, and in many ways still uncharted. But the fragments speak. From chemical traces in deep-sea black shales, from rare Lagerstätten where soft tissues were preserved against the odds, and from modern deep-sea biology, which offers living relics of ancient lineages still persisting in the abyss, scientists have assembled a picture of prehistoric deep-sea life that is both scientifically rigorous and genuinely astonishing. The deep Silurian Ocean, for example, roughly 420 million years old, was home to communities of organisms that are now known only from a handful of fossil localities. Among these were the Stylophora, a bizarre group of echinoderms with body plans so unusual that their precise relationship to other animal groups was debated for over a century. There were also early representatives of the Polychaete worms, segmented marine worms that today represent one of the most successful and diverse animal groups on Earth, numbering over 10,000 known species, living in the deep sediments and processing organic material in ways that profoundly influence the chemistry of the overlying water column. The late Devonian Ocean, roughly 370 million years ago, is of particular scientific interest because it coincided with one of the most severe mass extinctions in Earth's history, the Kellwasser and Hangenberg crises, collectively known as the Late Devonian Extinction. Approximately 75% of all marine species disappeared over roughly 20 million years, with the most severe pulses compressed into intervals of 100,000 to 200,000 years. The deep ocean during this period showed evidence of severe anoxia linked to rapid climate change, sea level fluctuations, and terrestrial organic matter entering the ocean as the first land plants began altering river chemistry across the continents. The work of Dr. Paul Wignall at the University of Leeds, who has spent decades analyzing the sedimentary record of mass extinction events, has been instrumental in documenting the deep ocean signature of these crises. His research has shown that deep-sea anoxia was not simply a consequence of extinction events. In many cases, it was a driver. The expansion of oxygen-depleted zones from the deep ocean upward into shallower waters killed vast numbers of organisms that had no evolutionary response to a world without breathable water. And yet, each time, life in the deepest zones persisted. Not all of it, not unchanged, but the deep ocean, that vast, cold, dark, pressurized refuge, consistently served as a reservoir of biological diversity that outlasted every catastrophe above it. It is not merely a place where strange things live, it is a sanctuary, a vault, a place where the biological experiments of earlier ages are stored, modified, and occasionally, under the right circumstances, released back into the wider ocean when conditions at last allow. It is a library written in living tissue, and it has been accumulating entries for over 500 million years. One of the most compelling modern demonstrations of this principle involves a group of organisms known as coelacanths. The coelacanth, Latimeria chelumnae, was known to science only from fossils when, in 1938, a living specimen was discovered in a trawl net off the coast of South Africa by museum curator Marjorie Courtenay-Latimer. The fish was identified by ichthyologist J.L.B. Smith as a member of a group thought to have gone extinct 66 million years ago at the end of the Cretaceous period. It had survived in the deep submarine canyons of the western Indian Ocean, utterly unknown to modern science, for 66 million years. It is a living fossil, a survivor, a message from the deep to the surface, written in flesh and fin. The coelacanth is not unique in this regard. The nautilus, a living relative of those ancient orthocerids we encountered at the opening of this journey, has persisted largely unchanged for over 400 million years. The horseshoe crab, Limulus polyphemus, has been present in essentially its current form for at least 450 million years. The priapulid worms, small, unsegmented marine predators that appear bizarre even by modern standards, have a fossil record extending back to the Cambrian over 500 million years ago, and they still live in the muddy sediments of cold, deep marine environments today. These are not curiosities. They are evidence of a principle that defines the story of prehistoric oceans at every depth, that the deeper you go, the older the world becomes, and the more intact that ancient world remains. The science is clear. The deep ocean is Earth's longest-running experiment, its most persistent archive, and, as we are about to discover, its most extraordinary world. Let us descend now. Not in the abstract language of science, but in the full sensory reality of what these ancient oceans were, what they looked, felt, sounded, smelled, and tasted like, as best as the fossil record and our understanding of ancient ocean chemistry allows us to reconstruct them. We begin at the surface. The Ordovician Ocean, that vast, warm, amber-lit sea of 480 million years ago, had a surface temperature in its tropical zones of approximately 40 degrees Celsius. Not a comfortable temperature by any standard. The water was warm enough that you would feel it as heat against your skin, like the water of a shallow tropical lagoon amplified by a factor that would make you immediately want to leave. It was also significantly saltier than modern seawater, with dissolved mineral concentrations reflecting a world where the balance of continental weathering, volcanic outgassing, land-ocean chemistry had not yet settled into the patterns of the modern era. The sky above this ocean was different in ways that went beyond temperature. The carbon dioxide concentration of the early Ordovician atmosphere was approximately 16 times higher than pre-industrial modern levels. This did not create a hellish, uninhabitable world. Complex life had already been thriving in these conditions for tens of millions of years. But it did mean that the ocean's ability to absorb and buffer carbon dioxide operated under entirely different rules. The water was more acidic than modern seawater. The rate of carbonate mineral dissolution was higher, and the creatures that built their shells and skeletons from calcium carbonate, which is to say, a very large proportion of the marine animals in these seas had evolved their chemistry specifically to cope with conditions that would severely stress their modern equivalents. At the surface, the Ordovician Ocean was not a quiet place. It was in constant motion, swept by currents driven by the configuration of the continents and the angle of the prevailing winds, which were themselves shaped by a planet with a different axial tilt and a slightly faster rotation than the Earth of today. A day in the early Ordovician lasted approximately 21 hours rather than 24, because the Moon was slightly closer to the Earth and its gravitational braking had not yet fully slowed Earth's rotation to its modern pace. The tides, driven by that closer Moon, were slightly stronger than those of the modern world, and their rhythm was fractionally faster. You stand at the water's edge, mentally, luxuriously, without danger, and let your senses adjust. The sound of the Ordovician Ocean is immediately strange. There are no seabirds. There have never been seabirds. There are no calls, no distant whale songs, no sounds of breaking shells on rocks. The dominant acoustic presence at the surface is the water itself, waves, currents, the deep, low percussion of oceanic motion transmitted through hundreds of meters of water. Below the surface, the sound world would have been profoundly unlike anything a modern diver hears. The biological soundscape of modern oceans, full of the clicks, grunts, chirps, and songs of fish, dolphins, crustaceans, and invertebrates, would be entirely absent in the Ordovician. What sounds existed were mechanical, the movement of hard shells through water, as the soft percussion of bodies colliding in the current, the almost inaudible rustle of tentacles and filtering appendages in constant motion. Beneath the surface layer, that upper 200 meters known as the photic zone, where sunlight still penetrates in sufficient quantity for photosynthesis. The Ordovician Ocean was populated by communities of organisms that modern biologists would find both recognizable in their general principles and utterly alien in their specific forms. Here, in the sunlit upper waters, drifted vast blooms of cyanobacteria, the ancient descendants of the microorganisms that had, over the preceding 2 billion years, gradually transformed Earth's atmosphere by producing oxygen as a byproduct of their photosynthesis. Cyanobacteria in the Ordovician formed long, untangled filaments and colonial mats that stretched for kilometers in the open ocean, their blue-green pigmentation giving the sunlit upper water a faint otherworldly tint. They were grazed upon by enormous swarms of small zooplankton, mostly early forms of crustaceans and other tiny arthropods, which in turn fed the filter feeders, the small cephalopods, and the drifting communities of graptolites. The graptolites deserve particular attention. These colonial organisms, whose name means written stone in ancient Greek, a reference to their appearance in fossils which resemble hieroglyphic marks on slate, were among the most abundant and widespread organisms in the Ordovician Ocean. A graptolite colony, called a rhabdosome, consisted of a series of individual animals called zooids, each housed in a tiny cup-shaped structure called a theca, arranged along a shared organic framework. Some graptolite species floated freely in the water column, suspended by a gas-filled float, a float that may have been as sensitive to depth as a modern submarine's ballast system, drifting with the current and catching food with tentacles that extended from each tiny cup. Other species were benthic, anchored to the seafloor, their branches extending upward into the current like the arms of a miniature tree. A single graptolite rhabdosome was rarely more than a few centimeters in length, but in the Ordovician Ocean they occurred in densities that are almost impossible to visualize today. Our geological samples from Ordovician age deep water shales, particularly from the Skiddaw group in the English Lake District and from equivalent formations in Wales and Scandinavia, contain graptolite fossils in such profusion that, in life, the water column in certain zones must have been almost curtained with their drifting colonies. They were a defining feature of the Ordovician Ocean's visual landscape, and their fossils have become one of the most important biostratigraphic tools in geology, used to date rocks and correlate sedimentary sequences across continents, because they evolved rapidly and spread globally, leaving a sequential record of forms that changed from one geological moment to the next. Deeper now, below 200 meters. The transition into the dysphotic zone, the zone of twilight, where light is present but insufficient for photosynthesis, is gradual in the modern ocean and was gradual in the Ordovician too. But the experience of it is not gradual. It is a slow erasure. The warm amber light of the surface gives way to a deeper, cooler blue. The temperature drops, not sharply, but persistently, several degrees for every additional 100 meters of depth. The pressure increases at a rate of approximately 1 atmosphere per 10 meters, meaning that at 300 meters depth, you are experiencing 31 atmospheres of pressure pressing on every surface of your body. At 500 meters, 51 atmospheres. At 1,000 meters, 101 atmospheres. The physics of pressure in the ancient ocean were identical to those of the modern ocean because pressure at depth is a function of water density and gravity, neither of which has changed meaningfully in the past half-billion years. In the Ordovician twilight zone, at depths of roughly 200 to 600 meters, lived communities of organisms whose ecology was shaped by a principle that has remained constant across all of geological time. In the absence of sunlight, food must come from above. Dead and dying organisms from the surface, phytoplankton, zooplankton, graptolite colonies, fragments of larger creatures, sank slowly through the water column, degrading as they fell, stripped of their energy content by the bacteria and small predators that intercepted them on the way down. What reached the deeper zones was not the rich organic material of the surface, but a diluted, partially decomposed rain of organic particles, marine snow, modern oceanographers call it, that drifted slowly downward in continuous ghostly curtains. This marine snow was the foundation of life in the deep Ordovician Ocean. The organisms that lived at depth in the Ordovician were adapted to intercept it, concentrate it, and extract maximum energy from minimum material. Among the most important of these were the early echinoderms, not the starfish and sea urchins that dominate modern mental images of this group, but their ancient and stranger relatives. The carpoids, for example, were flattened, asymmetrical echinoderms with a bizarre, lopsided body plan that has made their classification a subject of intense scientific debate for over a century. They clung to the seafloor or to rocky substrates in the deeper water, using their flexible appendages to capture the organic rain drifting downward from above. Their forms were wildly diverse. Some resembled flattened bags studded with calcite plates. Others bore a passing resemblance to nothing in the modern world whatsoever. Alongside them lived communities of sponges, not the familiar bath sponge of modern commerce, but the archaeocyaths and their descendants, some of which had already begun to form the first proto-reef structures in the shallower parts of the ancient seafloor. In deeper water, the sponge communities were more diffuse, their individuals larger and their filtration systems more specialized for the lower particle concentrations of the deep. And then there were the large predators. In the deeper water of the Ordovician, perhaps most menacing of all were the nautiloids. Not a single species, but an entire radiation of cephalopod diversity that, by the Middle Ordovician, had produced both the coiled nautiloid form recognizable from the modern nautilus and the straight-shelled orthocone form, the Orthoceras type, that we first encountered at the surface. The largest Ordovician nautiloids were genuinely massive animals. Orthoceras, a genus of orthocone nautiloid from the late Ordovician, may have reached lengths of 6 to 9 meters. Estimates vary because complete specimens have never been found. If those upper estimates are correct, Orthoceras was among the largest animals alive on Earth at that time, a slow-moving, jet-propelled ambush predator whose shell alone would have weighed several hundred kilograms, propelling itself through the water column with bursts of expelled water, hunting by chemical sense and primitive vision in the cold, dim depths. Imagine one of these creatures drifting at 300 meters below the surface of the Ordovician Sea. Its shell gleams faintly in the near-total darkness, just enough residual bioluminescence from the organisms it has recently disturbed to catch the faint tracery of its spiral chambers. Its tentacles extend forward in a wide, searching fan. Its eye, a large camera-type eye, remarkably similar in basic structure to the eyes of modern cephalopods, a structure that evolved independently from vertebrate eyes and represents one of evolution's most striking examples of convergence. Turns slowly, scanning the darkness for movement. And in the silence and the cold and the dark, something moves, something smaller. A trilobite, perhaps. In navigating the deep sea floor on its 14 pairs of legs, its compound eyes detecting the chemical signature of the nautiloid's approach just a fraction of a second too late. Now, go deeper still. Below 1,000 meters into the Bath hypelagic zone, which modern oceanographers define as the region between 1,000 and 4,000 meters. The Ordovician Ocean entered a realm of complete, permanent darkness. Not the soft twilight of the mesopelagic, not the dim blue filtering of the dysphotic zone. True, absolute darkness. The kind of darkness that has a texture, not merely the absence of light, but a presence, a weight, a uniformity that the human sensory system, built for a world of sunlight and shadow, struggles to process meaningfully. In this darkness, the temperature of the Ordovician deep ocean was cold, very cold. Studies of oxygen. Isotope ratios in Ordovician deep water carbonates suggest bottom water temperatures in the range of 10 to 15 degrees Celsius during the early and middle Ordovician, warmer than the modern deep ocean, where bottom waters in most basins hover near 2 to 4 degrees Celsius, but still profoundly cold relative to the sun-warmed surface. As the Ordovician progressed toward its close, culminating in the late Ordovician mass extinction of approximately 445 million years ago, triggered by a rapid glaciation event centered on the southern supercontinent of Gondwana, bottom water temperatures would have dropped dramatically as cold, dense glacial meltwater poured into the ocean basins and displaced the warmer, older deep water. But before that catastrophe, in the warm, anoxic basins of the deep Ordovician Ocean, a particular category of organisms thrived that deserves special mention: the anaerobic microbial communities. These were communities of bacteria and archaea, the most ancient and most fundamental categories of cellular life, that had adapted to an environment entirely without oxygen, deriving their energy from the chemical reactions of sulfur compounds, methane, and other reduced molecules present in the sediment. They formed dense, complex mats on the seafloor, processing organic material that had sunk from the upper ocean and releasing chemical compounds that in turn shaped the chemistry of the overlying water. In the deepest basins, where water circulation was poorest and oxygen had been depleted by the slow bacterial decomposition of accumulated organic matter, these microbial communities were the dominant form of life. Not exotic edge cases, the dominant life form. This was the world below the Ordovician Ocean, a world of darkness and cold, of chemical gradients and organic snow, of giant nautiloids drifting through the midwater and trilobites picking their way across the ooze-covered floor, of graptolite colonies slowly sinking as their float collapsed, of microbial mats pulsing with the slow chemistry of a world without oxygen. A world that was, in the truest sense, ancient even in its own time, because the microbial communities of those deep basins carried within their biochemistry the legacies of organisms that had first evolved over 2 billion years earlier, in the oceans of a world so different from the Ordovician that comparison strains the limits of language. And this world, this deep, dark, ancient world, was not static. It was dynamic. It was evolving. It was in the process of becoming something that, 480 million years later, we would be able to recognize in fragments and echoes in the organisms of our own modern deep sea. The story of what happened next, how the deep ocean responded to the events above, how life in the depths survived the catastrophes that repeatedly reset the surface world, and how those deep refugia shaped the course of evolution for every creature that followed, is a story that takes us on a journey through deep time that is both scientifically extraordinary and, in its own quiet way, profoundly beautiful. We have only just begun to descend. And here we pause, not to rest, not to summarize, but to stand at the edge of a question that has been building beneath everything we have explored so far, a question as deep and as dark as the ocean floor itself. We have seen the surface of the prehistoric ocean. We have watched the nautiloid hunt in the middle water. We have descended through the twilight zone and into the permanent darkness of the deep. We have glimpsed the microbial mats pulsing in the anoxic sediment of the Ordovician abyss. And at every stage of this descent, one observation has repeated itself. The deeper we go, the stranger the world becomes. But what does stranger really mean here? It is easy to say that deep-sea creatures are strange. Modern deep-sea biology is full of images and descriptions that seem almost too extreme for reality. The anglerfish, with its bioluminescent lure dangling above jaws that open wider than its own body, the barrelly fish with its transparent head and upward-facing tubular eyes, the giant isopod, the vampire squid, the zombie worm, the fangtooth. These animals populate the imagination with a kind of delighted horror. They are strange in the way that a funhouse mirror is strange, familiar elements combined in unexpected proportions. But the strangeness of the prehistoric deep ocean is different in kind, not just degree. It is the strangeness of a world that operated by different rules, not metaphorically different, chemically, physically, biologically different. The deep Cambrian Ocean had oxygen levels measured in fractions of a percent. The deep Cretaceous Ocean periodically lost oxygen altogether across entire basins for periods of time measured not in centuries but in hundreds of thousands of years. The deep Silurian Ocean held chemical gradients so steep, the difference between the oxidized surface water and the anoxic deep water concentrated into a zone of just a few hundred meters, that they created evolutionary pressures of an intensity we can barely model with modern tools. These are not slightly unusual conditions. These are alien conditions by the standards of the modern world, and yet life thrived in them. And here the question deepens. If the deep prehistoric ocean was populated by life forms adapted to conditions that make our own deep sea look comparatively hospitable, if those creatures evolved in environments of extreme pressure, extreme chemistry, and extreme darkness and extreme time, what did that do to them? Not just physically, not just in terms of body plan or metabolic strategy, but evolutionarily. Over the deep time of hundreds of millions of years, what did living in those depths do to the trajectory of life on this planet? Because here is the thing that modern science keeps discovering, quietly and with increasing confidence, about the relationship between the deep ocean and the rest of life on Earth. The deep ocean is not a passive participant in the history of life. It is an active driver. When the surface ocean is reset by a catastrophe, an asteroid strike, a volcanic winter, a rapid climate change, the deep ocean serves as the reservoir from which recovery begins. The organisms that survive in the deep, protected by their depth from the worst effects of whatever has gone wrong at the surface, are the seeds from which new surface communities eventually grow. This process has happened at least 5 times in Earth's history at the scale of global mass extinction and countless more times at smaller regional and local scales. The deep ocean has launched life back into the sunlit world over and over again. But each time it does, the life it sends back is shaped by what happened in the deep during the interval of catastrophe. It has been modified by the pressures of the deep environment. It carries the marks of its refuge in its biochemistry, its physiology, its behavior. It is not the same life that disappeared into the dark. It is something new, shaped by the deep, carrying the deep's ancient chemistry and its ancient adaptations into a changed and unfamiliar surface world. This is why the story of prehistoric oceans gets stranger the deeper you go. Not because the deep is unusual in isolation, but because the deep is the place where the oldest rules still apply, where the chemistry of 2 billion years ago still shapes what is possible, where evolution operates on a timescale so vast that what looks like stasis from the outside is, in fact, the slowest and most profound form of change imaginable. The questions that follow from this are layered and extraordinary. What specific lineages survived in the deep during each of Earth's major extinction events? What did they carry from those ancient refugia when they eventually returned to the surface? And what does the pattern of deep ocean survival tell us about the future? about what is happening in the deepest basins of the modern ocean right now as the surface world undergoes changes that, by any geological measure, qualify as rapid and severe. Those are the questions we will now explore. But first, a moment of stillness. A pause at the threshold, because we are about to enter the speculative spiral, the place where verified science gives way to informed extrapolation, where the known facts become the seeds of a larger story, and where the full, strange, magnificent complexity of the prehistoric deep ocean begins to reveal itself. Take a breath. Settle deeper into wherever you are. The darkness below is waiting. But here is where the story gets truly strange. And I want you to stay with this, because it is the kind of information that does not merely surprise you. It reframes everything you thought you understood about when life became complex on this planet. In 2018, a team of researchers led by Dr. Abigail Dodd at the Australian National University published a study in the journal Science Advances that analysed molecular biomarkers, chemical traces of biological activity preserved in ancient rock, from 1.6 billion-year-old marine sediments found in central India. These sediments were not from the Cambrian or the Ordovician or even the Precambrian shallows where most early life research is focused. They were from a deep marine basin laid down in still oxygen-depleted water more than 1.5 billion years ago, and they contained the molecular signature of a red alga. Not a bacterium, not an archaea. A eukaryote, a complex nucleated cell with membrane-bound organelles, an organism that, by everything previously understood, should not have existed for at least another 200 million years. This finding, if it holds up to continued scrutiny, and subsequent studies have not contradicted it, would push the origin of complex eukaryotic cells back to at least 1.6 billion years ago. It would mean that the deep ocean environments of the Middle Proterozoic, those ancient, warm, stratified, largely anoxic basins that we used to think of as lifeless or only bacterial, were in fact the cradle in which the first experiments in complex cellular life were conducted. The deep ocean was not merely a refuge for complex life during the Cambrian and after. It may have been the birthplace of that complexity in the first place. Think about what that means. Every complex organism on Earth today, every fish, every insect, every tree, every mammal, every human being, may owe the existence of its fundamental cellular architecture to experiments that happened in the dark, anoxic depths of an ocean that no longer exists. In water so old that the continents arranged above it bear no resemblance whatsoever to the geography of the modern world. In conditions so extreme that modern biology would classify them as marginal at best. The deep ocean did not merely shelter life, it may have invented it. Or rather, and this is the more precise and the more extraordinary formulation, it may have invented the kind of life from which everything recognizable eventually arose. The jump from simple bacterial chemistry to complex nucleated organelle-bearing cells is arguably the single most important transition in the history of life on Earth. It is the jump that made multicellularity possible. It is the jump that made animals possible. It is the jump that made us possible. And it may have happened in the dark, in the cold, in the deep, in an ocean so ancient that it was already old when the first trilobite crawled across the Cambrian seafloor. An ocean that no geologist has ever visited, that no instrument has ever sampled, that no camera has ever illuminated. An ocean that exists now only as chemical whispers in billion-year-old rock. That is where we came from. And the prehistoric oceans we have been exploring tonight, the Ordovician, the Cambrian, the Cretaceous, these are, in the grand sweep of deep time, not the ancient beginning of the story. They are the middle chapters. The early ones are older still, darker still, stranger still. The descent has no true bottom. CTA 2. If this story is carrying you somewhere peaceful, feel free to close your eyes. The narration will guide you. And so we begin the descent in earnest. Not a physical descent, though we will track the ocean's depth as a guide. Not merely a scientific descent, though the science will remain our compass throughout. This is a descent through time itself, a layered unwinding of the prehistoric deep ocean story, told in the sequence of its own unfolding, from the warm Ordovician waters we have already visited, downward and backward through the deepest corridors of Earth's oceanic history. We start not at the bottom, but at the first great turn. The first moment when the deep ocean demonstrated, in terms that the fossil record preserved with shocking clarity, that it was playing a different game from everything above it. That moment is 445 million years ago, the late Ordovician mass extinction. The extinction begins at the surface. It always does. Over the southern pole, the supercontinent of Gondwana has drifted into a position where its enormous landmass, comprising most of what will one day be the southern hemisphere's continents, begins to trap cold air. Ice begins to accumulate. Not the gradual accumulation of the modern ice ages, which operate on cycles of roughly 100,000 years, driven by subtle variations in Earth's orbit, but a faster, more severe glaciation, triggered by a combination of tectonic positioning and declining atmospheric carbon dioxide. Over approximately 500,000 years, a geological eye blink, the ice sheets grow until they cover millions of square kilometers of the Gondwanan landmass, trapping enormous volumes of water that had previously been circulating in the global ocean. Sea levels drop rapidly by geological standards. The shallow, warm tropical seas that had covered 85% of the continental shelves begin to drain. The communities of trilobites, brachiopods, graptolites, and echinoderms that had spent millions of years colonizing those shallow seafloors suddenly find their habitat contracting beneath them. The warm, well-oxygenated waters retreat. The carbonate reefs that had provided structural complexity and ecological niches for thousands of species are exposed to air and die. The temperature of the surface ocean drops by an estimated 10 to 15 degrees Celsius over this interval. This would mean that the surface ocean communities, those extraordinarily diverse assemblages that had taken nearly 50 million years to build up through the Ordovician radiation, were exposed to a set of environmental changes that they had no evolutionary experience with and no physiological preparation for. And approximately 85% of marine species disappear from the fossil record across this interval. Not immediately, not in a single catastrophic moment, but in pulses, waves of extinction that struck different depths, different latitudes, and different ecological communities at different times within the broader crisis. Now watch what happens in the deep. The deep ocean during the early phases of the late Ordovician extinction was already a stressed environment. The shift toward a glaciated world had changed the pattern of deep water formation. Cold, dense water was being generated at the glaciated southern pole and sinking to the bottom of the ocean, carrying with it oxygen from the surface and pushing the older, warmer, oxygen-depleted bottom water upward and outward in a circulation reversal that poisoned shallow water communities with anoxic water from below. This is a process modern oceanographers call upwelling, and when it is driven by deep water circulation changes rather than simple wind-driven coastal dynamics, it can introduce severely oxygen-depleted water into shallow coastal environments where organisms are utterly unprepared for it. The combination of cooling, sea level fall, and anoxic upwelling created a crisis that was multidirectional and almost impossible to escape at moderate depths. But at great depth, below 2,000 meters in the abyssal basins far from the continental shelves, the organisms that had already adapted to low oxygen, extreme pressure, and darkness experienced the extinction differently. Not as catastrophe, but as change. Change they were, in many cases, equipped to accommodate. Over the next 2 million years, as the glaciation reached its peak and then, with equal speed, retreated as volcanic activity continued to pump carbon dioxide into the atmosphere and warm the planet again. The deep ocean communities gradually reorganized. Species that had existed only at depth began to extend their range upward into the recovering shallow environments. New lineages, bearing the biochemical signatures of deep ocean adaptation, resistance to low oxygen, tolerance of high pressure, metabolic flexibility, began to colonize the ecological space left vacant by the extinctions above. This would mean that the Silurian Ocean, the geological period that followed the Ordovician, beginning approximately 443 million years ago, was not simply a diminished version of the Ordovician. It was a remixed world. Its shallow water communities were partly rebuilt from survivors that had weathered the extinction in deeper water, bringing with them adaptations forged in the dark and the cold. The Silurian reef communities, built primarily by tabulate and rugose corals, stromatoporoids, and brachiopods, showed different species compositions from their Ordovician predecessors, and modern analysis of their physiology suggests they were somewhat more tolerant of environmental variability, a characteristic consistent with deep ocean ancestry. The story doesn't end there. Over the next 30 million years, through the Silurian and into the Devonian, the age of fishes, the ocean underwent a transformation as dramatic as any in its history, but driven not by catastrophe this time, but by evolutionary innovation. The Devonian Ocean, roughly 419 to 359 million years ago, was the ocean in which vertebrates first became a major ecological force. Fish, which had existed as minor players in the Ordovician and Silurian, underwent a massive evolutionary radiation in the Devonian, diversifying into hundreds of forms that occupied ecological niches across every depth and every habitat type. The placoderms, armored fish with bony head shields and razor-sharp jawbones derived from gill arches, became the dominant large predators of Devonian seas. The most famous placoderm, Dunkleosteus terrelli, reached lengths of up to 8 or 9 meters and possessed the most powerful bite force of any fish in Earth's history, estimated at approximately 5,000 newtons at the tips of its shearing jaw plates, equivalent to roughly 500 kilograms pressing on a single point. Dunkleosteus hunted in the upper water column, preying on large fish, cephalopods, and smaller placoderms. It was a surface world predator, shaped by the evolutionary pressures of the photic zone. But deep below it, in the Devonian twilight, and below, a different evolutionary story was unfolding. The early sharks, members of the class Chondrichthyes, with the cartilaginous fish, first appeared in the early Devonian approximately 420 million years ago, and almost immediately began to explore deep water environments. Unlike the placoderms, which were heavily armored and relatively inflexible in their ecology, the early sharks were lightweight, fast, and metabolically adaptable. Their cartilaginous skeletons, which are less dense than bone, gave them a natural tendency toward neutral buoyancy at depth. Their electrosensory systems, the ampolli of Lorenzini, a series of pores on the snout that detect the electric fields generated by the muscular contractions of other animals, made them extraordinarily effective hunters in conditions of low light and zero visibility. The deep Devonian Ocean was a natural environment for sharks, and the fossil record, as fragmentary as it is for deep-water Devonian sediments, suggests they made use of it. Over the next 30 million years, through the Devonian and into the Carboniferous period, the deep ocean continued its role as both refuge and innovator. The sharks diversified into forms that are almost unrecognizable to modern eyes, not the sleek, torpedo-shaped predators of popular imagination, but strange, flat-bodied, spiny, multi-finned creatures that explored an extraordinary range of body plans in what paleontologists sometimes call the Shark's Dark Age, a period when their evolutionary experimentation was at its most radical and their fossil record at its most ambiguous. And then, 10,000 years after the late Devonian crisis reached its most severe phase, more precisely, during the Hagenberg event of approximately 359 million years ago, which closed the Devonian period, the ocean reset again. The Hagenberg event was an extinction of particular severity for marine vertebrates. The placoderms, which had dominated the Devonian Ocean for 60 million years, disappeared entirely. Gone. Not reduced to a few remnant lineages. Gone. Every species of every placoderm genus, from the tiny jawless forms that had preceded them to the enormous Dunkleosteus, vanished from the fossil record at the Devonian Carboniferous boundary and never appeared again. It was a complete extinction of one of the most successful vertebrate groups Earth had ever produced. The sharks survived. Not all of them, not unscathed, but they survived. And the pattern of their survival is instructive. The species and lineages that made it through the Hagenberg were disproportionately those with deep water ecological connections, those that had access to the refugia of the bathypelagic zone, where the Hagenberg's combination of anoxia and rapid sea level change was less immediately lethal than in the shallow water environments where the placoderms had been most dominant. This would mean that the Carboniferous Ocean, a world of warm, shallow tropical seas beneath a sky thick with oxygen, a world of the vast coal swamps on land, and the first true reefs in shallow water, was rebuilt in its vertebrate ecology from the deep. The sharks that repopulated the surface after the Hagenberg carried the genetic and physiological legacy of deep ocean survival. They were, in a meaningful sense, our creatures of the deep returned to the light, and they thrived. Over the next 100 million years, through the Carboniferous and the Permian, the sharks diversified into hundreds of species, colonizing shallow and deep environments alike, developing body forms and hunting strategies of extraordinary variety. The Helicoprion, a Permian shark whose spiral whorl of teeth, arranged in a continuous coil, has no modern equivalent and whose precise anatomy was the subject of intense scientific debate for over a century, explored deep water environments and fed on soft-bodied cephalopods with its bizarre, circular saw of a jaw. The Stethacanthes, an earlier Carboniferous form, bore a unique anvil-shaped dorsal fin structure topped with denticles that match the denticles on the top of its head, a structure whose function remains unclear to this day, but whose sheer biological inventiveness speaks to an evolutionary system operating in conditions of radical experimentation. Now the story reaches its most profound turn. 252 million years ago, the end Permian mass extinction, the Great Dying. This is the largest mass extinction in Earth's history. Not just in scale, approximately 96% of all marine species and 70% of terrestrial vertebrate species were extinguished, but in mechanism. The Great Dying was driven by a catastrophic episode of volcanic activity centered on the Siberian Traps, a vast volcanic province in what is now Siberia, which erupted continuously for approximately 1 million years, releasing staggering quantities of carbon dioxide, sulfur dioxide, and other volcanic gases into the atmosphere. The carbon dioxide warming drove global average temperatures up by approximately 10 to 12 degrees Celsius over the course of roughly 60,000 years, the most rapid sustained warming event in Earth's geological record. The sulfur dioxide formed acidic aerosols in the atmosphere, killing photosynthetic organisms on land and in the ocean. Ocean acidification, the lowering of seawater pH as excess carbon dioxide dissolved into the surface, reached levels that made carbonate shell building impossible or extremely difficult for most marine invertebrates. Ocean deoxygenation, the depletion of dissolved oxygen from warming water and disrupted circulation, spread from the deep ocean upward into the mesopelagic and photic zones. The ocean effectively suffocated. At the surface, the impact was near total. The extraordinary diversity of Permian marine life, the brachiopods, corals, fusulinid foraminifera, trilobites, already reduced by this point, blastoids, and dozens of other groups, collapsed. The trilobites, which had been a presence in Earth's oceans for nearly 300 million years since the base of the Cambrian, disappeared entirely, never to return. The corals that had built the Permian reefs were almost entirely gone. The reefs themselves, those complex three-dimensional structures that had hosted thousands of species for tens of millions of years, collapsed to rubble and did not recover for approximately 8 to 10 million years. In the deepest ocean, the story was more complex. The abyssal sediments of the early Triassic, the period immediately following the end Permian, show a characteristic pattern that paleoceanographers have studied intensively. A prolonged interval of carbon isotope disruption, called the early Triassic carbon isotope excursion, that lasted for approximately 5 million years and reflected a profound disorganization of the global carbon cycle. The deep ocean was anoxic throughout this interval in many basins. Its benthic communities, the organisms living on and in the seafloor, were reduced to microbial mats and a handful of stress-tolerant animals, mostly small, simple organisms with low metabolic demands and high tolerance for chemical extremes. But some things survived. It was among the most scientifically important survivors of the end Permian deep ocean were the lineages of bivalve mollusks, specifically the deep water bivalve groups related to the modern genera Nucula and Nuculana that had occupied deep-sea sediments since at least the late Paleozoic. These organisms, small and anatomically unspectacular, were among the primary colonizers of the Triassic deep-sea floor in the aftermath of the extinction. Their presence in early Triassic deep-water sediments, documented in studies from localities in China, Austria, and Canada, provides direct evidence of benthic community persistence in the deep ocean across the extinction boundary. And so, 5 million years after the Great Dying reached its most catastrophic phase, in the Middle Triassic, roughly 247 million years ago, as the global carbon cycle slowly stabilized and ocean circulation patterns began to re-establish, the process of biological recovery began to draw upon those deep refugia. New groups emerged to fill the ecological space left vacant by the mass extinction. The Mesozoic Marine Revolution, a term coined by paleontologist Geert Vermeij to describe the dramatic escalation of predator-prey interactions in Mesozoic marine communities, drew partly on lineages that had survived the Great Dying in the deep, carrying their deep ocean adaptations into a recovering world of extraordinary ecological opportunity. Among the new occupants of the Triassic and Jurassic Ocean was a group of reptiles that would become the most perfectly adapted ocean predators of the entire Mesozoic, the ichthyosaurs. The ichthyosaurs appeared in the fossil record in the earliest Triassic, perhaps within 2 million years of the end Permian extinction, already as fully marine animals, a pace of marine adaptation so rapid that it remains one of the most remarkable evolutionary transitions in the vertebrate record. They were not fish, they were reptiles, descended from land-dwelling ancestors that had returned to the ocean and within a geologically brief interval had evolved a body plan of extraordinary hydrodynamic efficiency, fish-like in silhouette with a crescent-shaped tail, large stabilizing flippers, and a streamlined skull bearing numerous conical teeth suitable for capturing fish and cephalopods. Their large eyes, in some species the largest eyes of any vertebrate ever to have lived, the reaching diameters of 26 centimeters in the species Temnodontosaurus platyodon, suggest an emphasis on vision in low-light conditions, leading researchers to hypothesize that the large-eyed ichthyosaurs were adapted for deep, twilight zone hunting, diving to depths where the cephalopods they preferred were most abundant. The ichthyosaurs, in other words, were creatures of the surface world that had learned the tricks of the deep, following a path toward the abyss that evolution had opened once before for the sharks and would open again for the whales tens of millions of years later, a recurring evolutionary invitation extended from the deep ocean to the surface world, accepted again and again across geological time. Over the next 80 million years, through the Jurassic period, roughly 201 to 145 million years ago, The Mesozoic Ocean became one of the most ecologically complex marine worlds Earth had yet produced. The dominant groups were the ammonites, coiled cephalopods that had survived the end Permian in reduced form and subsequently diversified into thousands of species of extraordinary variety. Their shells ranging from tightly coiled spirals, the size of a coin, to loosely uncoiled forms the diameter of a cartwheel. And the belemnites, squid-like cephalopods with solid internal guards that accumulated in the sediment record in such numbers that their fossils, called guard fossils or thunderbolts by medieval Europeans who did not know what they were, can be found by the handful in Jurassic Age rock exposures across Europe, Africa, and the Americas. The Jurassic Deep Ocean was not merely a backdrop for this surface drama, it was a participant. The sediment record from Jurassic deep water environments, particularly from localities in the English Midlands, from the Posidonia Shale of southern Germany, and from equivalent formations in France and Switzerland, shows a bottom water environment that oscillated between oxygenated and anoxic conditions on timescales of tens to hundreds of thousands of years, driven by changes in ocean circulation related to the opening of the Atlantic Ocean as the supercontinent Pangaea continued its slow fragmentation into the familiar continental arrangement of the modern world. These oscillations created a pulsed pattern of benthic community establishment and collapse that drove evolutionary innovation at a remarkable pace. Groups that could tolerate low oxygen conditions, such as certain families of deposit-feeding worms, early holothurians, sea cucumbers, and specialized microbial communities, expanded their ranges during anoxic intervals and were then partially replaced by more oxygen-dependent groups when conditions improved. Each cycle left behind a slightly different community from the one that preceded it. Each cycle wrote a new chapter in the deep ocean's biological archive, accumulating layer upon layer of evolutionary history in the compressed sediment of the ocean floor, awaiting the drills of future scientists who would not exist for another 150 million years. Among the extraordinary organisms of the Jurassic Deep is one that deserves special attention. Sarcocoma, a free-swimming crinoid, a filter-feeding echinoderm that occurred in such vast numbers in the Jurassic open ocean that its fragmented skeletal plates form thick layers in the Solnhofen limestone of Bavaria, the same formation that preserved the famous fossil of Archaeopteryx, the earliest known bird. Sarcocoma represents a lineage of deep water origin that had, by the Jurassic, expanded its range into the open ocean water column, exploiting the rain of phytoplankton from above with the same filtering strategy its ancestors had employed on the seafloor. It is a perfect example of the trajectory that defines the deep ocean's role in Earth's biological history. Lineages adapting in the deep, then expanding upward as conditions allow, carrying the innovations of the abyss into the sunlit world above. Now, 40 million years later, in the Middle Cretaceous, at approximately 100 million years ago, the Mesozoic Ocean reached what many paleontologists regard as its peak of ecological complexity and diversity. The seas were warm and expansive, their circulation driven by the equator to pole temperature gradient of a world without ice caps, their chemistry reflecting the enormous productivity of phytoplankton blooms in sunlit surface waters extending from the equator to near the polar regions. Plesiosaurs and mosasaurs, the great marine reptiles of the Cretaceous, dominated the upper water column as apex predators of extraordinary size and variety, from the long-necked, small-headed Elasmosaurus at roughly 14 meters in length to the barrel-bodied, enormous-jawed mosasaur Mosasaurus hoffmanni, which reached lengths of perhaps 17 meters and whose crushing bite could reduce ammonite shells to fragments. Below them, in the mid-water and twilight zones of the Cretaceous Ocean, the ammonites were reaching the apex of their evolutionary radiation, their forms becoming increasingly elaborate and, in many cases, increasingly strange. The heteromorph ammonites, a group that departed from the standard tightly coiled spiral to produce shells in the form of open spirals, irregular helices, U-shaped hooks, and even straight rods, represent an evolutionary experimentation with form that rivals anything in the Cambrian explosion for sheer improbable variety. The species Nipponites mirabilis bore a shell so convoluted and irregularly knotted that early scientists who encountered its fossils assumed it must be a pathological specimen, a deformed individual rather than a representative of an actual species. It was not. It was a successful, widespread, ecologically functional animal that apparently moved through the water column in its impossible-looking shell with perfect ease, guided by jet propulsion and a physiology tuned to an environment we can still only partially understand. And then, 66 million years ago, the asteroid arrived and the Cretaceous ended. The pattern that followed the Chicxulub impact in the deep ocean was the same pattern we have observed at every previous mass extinction boundary. Collapse at the surface, persistence in the deep, slow recovery from the bottom up. The nanoplankton, the microscopic photosynthetic organisms at the base of the marine food web, collapsed almost immediately after the impact, their extinction preserved in the sediment record as a sudden and dramatic disappearance of calcareous nanofossils at the Cretaceous-Paleogene boundary. The marine food web, deprived of its primary productivity base, experienced a cascading collapse that reached from the plankton through the zooplankton and small fish through the larger predators and ultimately to the apex marine reptiles that vanished entirely from the record at or near this boundary. But in the deep, the organisms that had already adapted to surviving on minimal food, the deposit feeders, the scavengers, the organisms that derived their energy from chemical reactions rather than from sunlight-derived organic material, were relatively less affected by the collapse of surface productivity. Their communities were disrupted, certainly, their diversity was reduced, but they were not erased. And over the next 10,000 years, as the atmosphere cleared and surface light returned, these deep-sea survivors began to seed the recovering surface world with the biological material for a new age. The Paleocene Ocean, the first chapter of the Cenozoic era, beginning 66 million years ago, was a strange and simplified world at the surface, its food webs rebuilt from scratch by a handful of surviving lineages. But at depth, so it retained a connection to the Cretaceous world that had just ended. The chemical signatures of Cretaceous deep water masses persisted in the abyssal basins for tens of thousands of years, gradually replaced as the new Paleocene circulation patterns became established, and the deep water was slowly turned over by the slow thermohaline circulation that carries cold, dense water from the polar regions to the ocean floor. In this new world, a remarkable evolutionary experiment was beginning in the shallow seas of the Tethys Ocean, the warm, equatorial seaway that stretched from what would eventually become the Mediterranean to the Indian Ocean. The early cetaceans, the ancestors of all modern whales and dolphins, were taking their first steps back into the water. Pachychaetus, a four-legged, wolf-sized mammal, lived approximately 53 million years ago in the shallow estuaries and rivers of what is now Pakistan, wading into the water to hunt fish with elongated jaws and teeth more reminiscent of a crocodile than of any modern whale. Within the following 15 million years, its descendants would complete one of the most dramatic evolutionary transformations in the history of vertebrate life, producing fully aquatic animals, the basilosaurids, that reached lengths of 15 to 20 meters and whose sinuous, elongated bodies moved through the warm Eocene ocean with the fluid grace of enormous marine serpents. The cetacean lineage was moving toward the deep, and the deep was waiting to receive it, as it had received every lineage that had ever been willing to make the descent. The sharks in the Devonian, the ichthyosaurs in the Triassic, with the plesiosaurs in the Jurassic, the mosasaurs in the Cretaceous, and now the whales in the Eocene and Oligocene, each group independently discovering the same evolutionary truth. that the deep ocean rewards the organisms willing to tolerate its conditions with access to resources and ecological space unavailable anywhere else on the planet. And so, over the next 30 million years, through the Oligocene and into the Miocene, the whales diversified. The mystichites developed their baleen plates and became the largest animals Earth had ever produced, their enormous bodies sustained by the extraordinary productivity of the cold, nutrient-rich waters of the expanding polar seas. The odontocetes, the toothed whales, evolved echolocation, a biological sonar system of such precision that it allowed them to navigate, hunt, and communicate in complete darkness at depths beyond the reach of any light, in waters colder and more pressurized than any surface world environment. The sperm whale, Physeter macrocephalus, perfected the dive to extraordinary depths, developing physiological mechanisms, collapsible rib cages, oxygen storage in specialized myoglobin-rich muscle tissue, the ability to tolerate the nitrogen narcosis that would incapacitate a human diver at a fraction of the depth, that allowed it to descend to at least 2,000 meters in pursuit of giant squid in the eternal darkness of the mesopelagic and bathypelagic zones. The story of the prehistoric deep ocean, from the Cambrian to the Cretaceous, from the Ordovician nautiloids to the Eocene basilosaurids, from the deep microbial mats of the Proterozoic to the echolocating sperm whales of the Miocene, is a story of continuous, unbroken biological engagement with the deepest environments on Earth. Each new group that descended into the deep carried the evolutionary innovations of its surface world ancestry into an environment that tested and refined those innovations in ways that the surface could not. Each group that survived in the deep through a mass extinction event carried the signature of that survival back to the surface when conditions allowed. Each cycle of descent and return left the ocean's surface communities slightly different from what they had been before, enriched in ways that are still being discovered by their connection to the ancient, patient, extraordinary world below. Let us dream now, not the sharp, active dreaming of curiosity and inquiry, but the slow, deep dreaming of deep time, the kind of dreaming that happens when you release the need to understand and simply allow yourself to be carried gently and completely by the immensity of what is, by the scale of what has been, by the quiet enormity of a world that existed in full, extraordinary, teeming reality for hundreds of millions of years before any consciousness arose to observe it, and that will continue to exist in its deepest parts long after the surface world has changed beyond recognition once again. Imagine that the late Ordovician mass extinction did not happen. Or rather, imagine that the glaciation came, as it came in the true history of the Earth, but that it was slightly less severe, that the ice sheets over Gondwana were smaller by perhaps 20%, that sea levels fell by 80 meters instead of 170, that the surface ocean cooled and many species struggled and some were lost, because evolution in any world always involves loss, but that the catastrophic, near-total collapse of the shallow water communities that defines the true Ordovician extinction did not occur. The thread was bent, but not broken. The experiment continued. In this alternate world, the Ordovician does not end in mass extinction. It transitions slowly and continuously into the Silurian. But a Silurian that is not a recovery world, not a world still reeling from the loss of 85% of its species and slowly rebuilding its communities from a handful of survivors. It is a Silurian that is a continuation. A world in which the extraordinary communities of the Ordovician deep ocean are simply the Ordovician deep ocean, continuing uninterrupted into a new geological chapter with all their diversity intact, all their ecological relationships maintained, all their evolutionary trajectories still in motion, developing forms and behaviors and interactions that we, in our world, can only imagine because the extinction cut that thread 445 million years before we arrived to look for it. What might those forms have been in the warm, deep waters of this unbroken alternate Ordovician? the nautiloids would have continued their evolutionary radiation without the disruption of the extinction. The Orthoceras lineage, those enormous, straight-shelled apex predators of perhaps 9 meters in length, might have developed further into animals of even greater sophistication, shaped by the relentless pressures of deep ocean predation across an uninterrupted span of millions of years. Not merely larger, not merely faster, but more neurologically complex, as the cephalopod lineage in our own world has consistently trended toward greater brain-to-body ratio and more elaborate behavior with every passing geological period, as the octopus and squid of the modern ocean demonstrate with their extraordinary problem-solving abilities, their tool use, their complex social behaviors, and their capacity for individual learning and memory that surprises every researcher who studies them. In the unbroken Ordovician, with 50 million more years of continuous deep-water occupancy without the reset of mass extinction, the nautiloid lineage might have arrived at a form of distributed intelligence on a schedule 400 million years ahead of the one our own world eventually followed. Not human intelligence, not even approximately human intelligence in its nature or expression, not the language-based, tool-using, socially organized intelligence of a primate species that evolved on a continent, but a distributed, chemosensory, behaviorally flexible intelligence shaped by the needs of a deep ocean predator navigating in darkness by chemical and magnetic signals, coordinating hunting strategies across hundreds of meters of water with others of its kind, to perhaps communicating through the modulation of bioluminescent signals, light produced by the animal's own body chemistry, flashed in patterns that encode information, that distinguish individual from individual, that carry something we might tentatively, carefully, humbly call meaning. In the deep, warm, chemically stable water of this alternate world, those nautiloid societies would have had an extraordinary environment in which to develop their cognitive complexity. An environment that, unlike the surface world, is not subject to the rapid weather changes, the seasonal variations in light and temperature, the constant visual complexity, and the perpetual threat of desiccation that drove the evolution of surface world intelligence. The deep ocean is, instead, a world of chemical gradients, pressure differentials, and three-dimensional spatial navigation, a world that might favor a very different kind of cognitive architecture from the one that eventually produced primates, shaped by very different selective pressures toward very different ends, but no less rich, no less complex, no less genuinely extraordinary for being utterly alien to everything we recognize as mind. Let this image settle into your mind as you breathe. The deep ocean, dark and vast and warm, alive with moving shapes that carry, somewhere in their ancient bodies, in the chemistry of their neurons and the complexity of their signal systems, the very beginning of something that might one day, in another million years or 10 million years of uninterrupted development, come to be called thought. Not thought as you know it, but thought as the ocean knows it, deep, slow, patient, unencumbered by urgency, shaped by the pressures of a world where the relevant timescale is not hours or seasons or lifetimes, but geological epochs. Now let the image change. Let it dissolve like mist in deep water, particles dispersing slowly in all directions until a new image forms in its place. Slower, darker, more distant. Let us move forward in time, not in the alternate Ordovician, but in our own world. Forward through the extraordinary complexity of the Silurian, the Devonian, the Carboniferous, and the Permian. Forward past the Great Dying and into the warm Triassic Sea. And here, in this forward projection, let us imagine that the end Permian extinction, catastrophic as it was, left behind in the deep ocean one lineage that in our own world did not survive. An early relative of the coelacanths, a lobe-finned fish of the deep Permian Sea, small and anatomically unspectacular and physiologically adapted over millions of years to the low oxygen conditions of the deep Permian basins, that in our world disappeared with the rest of the Permian marine fauna. But in this alternate thread clung on in an isolated deep basin somewhere in the Tethys Sea, protected by its depth from the worst of the anoxic upwelling and the surface temperature spikes, surviving the transition into the Triassic with its lineage intact and its evolutionary potential unspent. This survivor, let us call it, for the purposes of our imagination, a proto-Latimerian, an echo in deep water of what would one day become the coelacanth lineage, finds itself in the early Triassic in a world of extraordinary ecological opportunity. The great predators are gone. The trilobites are gone. The brachiopod reef communities are gone. The fusulinid foraminifera that had been a defining component of Permian shallow water ecology are gone. The deep basins are anoxic, yes, but this lineage has already adapted to low oxygen over millions of years of deep water occupancy, and it survives the transition with the quiet, unhurried persistence of a creature that has never needed speed to survive. Only patience, metabolic flexibility, and the deep ocean's own extraordinary capacity to shelter what the surface world cannot protect. And in the early Triassic, it begins to diversify. Slowly at first, cautiously. Then, as the ecological space opens further and the recovering world offers niches that have been vacant for millions of years more rapidly. Over the next 20 million years, through the Middle and Late Triassic, this lobe-finned lineage gradually colonizes the recovering shallow seas, its anatomy already adapted for slow and deliberate locomotion in the deep, proving surprisingly effective in the complex, three-dimensional structural environments of the first Triassic reefs, where the ability to navigate tight spaces and hover in place gives it access to prey that the faster, more streamlined actinopterygian fish cannot easily reach. The Jurassic Ocean in this alternate thread is subtly but significantly different from the one in our own history. The lobe-finned fish lineage, now diversified into perhaps dozens of species across multiple depth zones and ecological niches, has become a stable and important component of Jurassic marine ecology. Their distinctive locomotion, using muscular pectoral and pelvic fins as primary propulsors rather than the body and tail motion of the actinopterygians, has given them advantages in low-current, high-structure environments. Around the developing Jurassic reefs, they navigate the crevices and caverns with a fluid, precise movement that modern eyes, if they could see it, might find unexpectedly familiar. Not unlike the movement of a modern coelacanth in the submarine caves of the Comoro Islands, where living specimens of Latimeria chalumnae have been filmed at depths of 150 to 300 meters, hovering with slow, deliberate fin movements in the cold dark of a submarine cave, their eyes reflecting the light of the research submersible with a steady, ancient, unhurried calm. And in the deep Jurassic Ocean, in the bathypelagic darkness below 2,000 meters, the ancestral forms of the lineage persist, little changed from their Permian predecessors, moving slowly through the dark water and the continuous organic rain, their large reflective eyes gathering the faintest traces of ambient bioluminescence from the strange creatures that flash past them in the deep, their metabolism ticking over at a rate so slow that a single large meal can sustain them for weeks or months, their movements measured and deliberate and entirely without the urgency that characterizes the fast metabolizing fish of the surface world. This is the sleep induction zone now. The place in this journey where the world softens its edges and the images become longer and slower and less precisely defined, more dreamlike in quality. I'm more concerned with texture and atmosphere and the quality of light, or rather the quality of the absence of light, than with the specifics of species and geological periods and evolutionary mechanisms. Feel the weight of the deep water above you, pressing gently, uniformly on every surface. Not the sharp, invasive pressure of something bearing down from a single direction, but the total, encompassing pressure of a medium that surrounds you completely and applies its force equally from every point of the compass and every angle of the vertical, so that what you feel is not compression but containment, not constriction but embrace, the ocean holding everything within it with equal force, equal attention, equal ancient care, in the way that time itself holds everything that has ever happened within it, without preference, without urgency, without the particular kind of attention that requires an observer. The water here is cold, not the sharp, gasping cold of a winter plunge, not the cold that registers as alarm, that triggers the breath catch and the rapid heartbeat and the urgent body knowledge that this is dangerous and you must move. This is a different cold entirely, the cold of the deep ocean, which is a deep, pervasive, bone-settling cold that comes from a world where heat has not reached in tens of thousands of years, where the water circulating slowly along the seafloor entered the abyss somewhere in the polar regions, dense and heavy with the memory of a previous era's ice, and has been slowly migrating through the deepest basins ever since, touching the bottom mud with a patience that is indistinguishable at human timescales from permanence. The sound here is almost nothing, not quite nothing, almost nothing. There are the faint, low, unimaginably slow vibrations that travel through thousands of kilometers of water from distant ocean currents, from the movement of tectonic plates, from the breath of hydrothermal vents far across the basin floor, where superheated water, rich in minerals, erupts from the seafloor and creates entire ecosystems, powered not by sunlight but by the chemical energy of the Earth's own internal heat. Ecosystems that have been operating continuously for tens of millions of years and that represent, in the opinion of many astrobiologists, the most compelling model for life on other worlds that we possess. Worlds where sunlight never reaches but where the heat of an interior, combined with the chemistry of water and rock, creates the conditions for biological complexity in the permanent dark. There is also the almost inaudible movement of the Vampiroteuthis infernalis, the vampire squid, not truly a squid at all, but an ancient cephalopod that represents an evolutionary lineage so old it sits between the squids and the octopuses in the branching tree of cephalopod evolution, a living remnant of a Cretaceous deep-sea world preserved essentially intact in the oxygen minimum zone of the modern ocean, as it extends its long, delicate feeding filaments into the organic rain above it, collecting the slow-drifting particles of ancient biology in the absolute darkness with the unhurried precision of an animal that has had approximately 80 million years to perfect this particular technique. The Vampiroteuthis is one of the modern ocean's most extraordinary deep-sea inhabitants, and it is almost entirely unchanged from its Cretaceous ancestors. Living at depths of 600 to 1,200 meters, it inhabits the oxygen minimum zone. That band of oxygen-depleted water in the mid-ocean depths, where the decay of organic material from above creates conditions that most organisms cannot tolerate, but where Vampiroteuthis moves with complete ease, its hemocyanin-based blood, carrying oxygen at concentrations that would be impossible for most animals. Its entire physiology a masterclass in the biochemical innovations that the deep ocean has been developing for hundreds of millions of years. Its black-purple body, rarely longer than 28 centimeters from mantle tip to tentacle, is lined with photophores, bioluminescent organs that can be activated in different patterns and intensities to create a dazzling display of cold light in the darkness, used for communication, for camouflage, against the faint downwelling light from above, and for orientating itself in a three-dimensional world where all spatial reference points beyond the body itself have dissolved into a uniform, ancient dark. In the alternate Triassic-Jurassic world of our projection, the ancestors of the Vampiroteuthis exist alongside the Proto-Latimerian lineage in the deep basins of the early Mesozoic. They share the deep with a community of organisms that, in the dreamlike atmospheric quality of this long, slow, descending narrative, is both scientifically grounded in what we know of Mesozoic deep-sea ecology and imaginatively extended into what we might reasonably extrapolate. A community that includes the ammonites, whose coiled and uncoiled shells drift through the mid-water in every size and shape, moving slowly through their environment by jet propulsion, their septal chambers filled with gas at carefully regulated pressures that allow them to maintain neutral buoyancy at any chosen depth. Their tentacles extended in the dark to catch whatever small organisms drift within reach. And deeper still, below 3,000 meters on the abyssal plane of this ancient world, the microbial mats pulse and breathe in their ancient, unhurried chemistry, processing the organic material that has been raining down from the surface world for millions upon millions of years, are converting it into chemical forms that feed the larger organisms above them in the slow, circular, deeply patient economy of the deep. An economy that operates on timescales so different from anything at the surface that the difference is not merely quantitative but qualitative, not just faster or slower, but fundamentally different in kind, the way the deep ocean itself is different in kind from everything above it. There are communities on the abyssal plane of this imagined alternate Mesozoic world, and in the real abyssal plane of the modern ocean as well, that have been largely undisturbed for longer than most surface world communities last. Populations of deposit-feeding holothurians, sea cucumbers, ancient and slow, but their muscular bodies moving over the seafloor sediment so gradually that a camera placed to observe them would register their passage only through time-lapse photography. Their metabolisms tuned to a world where food is measured not in the rich, concentrated bursts of the surface, but in the slow, dilute, continuous fall of organic particles from above, each particle carrying its tiny cargo of ancient biology from a surface world the holothurian will never see and has no mechanism to imagine. The Paleodiction, that mysterious hexagonal pattern found in the sediment of some modern deep-sea floors, which may be the trace fossil of an organism that has never been conclusively identified, its maker unknown, its age uncalculated, its function undetermined, appearing in the geological record as far back as the Cambrian and still appearing on modern deep-sea photographs as a pristine geometric pattern in the soft mud of the ocean floor, is here too, as it has been here since before the Ordovician, as it will be here long after whatever civilization now exists at the surface has completed its brief geological appearance and dissolved back into the chemistry of the world. A pattern in the mud, a signature, a question mark written in the floor of the deepest ocean by something that has no name, in handwriting that has not changed in 500 million years. Lie back into this image, let it hold you. Let the details soften at the edges the way things soften in the hour before sleep, when the visual precision of the waking mind begins to give way to the looser, a more atmospheric mode of the dreaming mind, and what matters is no longer the specific facts. Which species, which geological period, which depth, which temperature? But the quality of the world being described, the texture of the silence, the weight of the dark, the slow and patient beauty of a place so old and so deep that it precedes every category the human mind has ever invented to describe it. The dark water above, the cold, still, pressurized world below. The slow movement of shapes in the darkness, the long deliberate drift of an ancient fish, the flash of a bioluminescent display in the middle distance, the quiet metabolic processing of the microbial mat, the slow accumulation of sediment, layer by microscopic layer, millimeter by millimeter. Each layer a compressed record of everything that happened in the ocean above during the centuries it took to be deposited, a record that will persist in rock for hundreds of millions of years beyond the moment of its formation, waiting for an intelligence capable of reading it that does not yet exist and may never exist, but that might, given enough time and the right circumstances, one day descend into these depths and find, in the chemistry of very old stone, the story of everything that the surface world forgot. Every layer tells a story. Every millimeter is a chapter. Every core extracted from the seafloor is a book, and the library is the entire ocean floor. And the library has been accumulating volumes since before the Cambrian, and most of it has never been read, and most of it never will be. And this is not a loss, but a feature. The ocean's archive is not meant to be read. It is meant to be kept, preserved. held in the cold and the dark and the pressure of the abyss with the patient, impersonal faithfulness of deep time, available to whatever comes next, whatever emerges next from the long, slow, unimaginably patient process of life, finding new forms for itself in a world that has been changing since its formation and will be changing still when the last ocean finally cools and stills and the last biological process runs down to its chemical conclusion and the world returns to the silence that preceded everything. But that is very far away, unimaginably far away. And tonight, the ocean is still warm and deep and alive. Tonight, the ancient world is still turning. The tides are still moving. The deep is still holding its archive. The strange creatures of the abyss are still drifting through the dark water, carrying their ancient biochemistry into another moment, another hour, another night like this one. Rest now in that knowledge. Let it settle into you the way sediment settles through deep water, not falling exactly, but drifting in the slow, unhurried spiral of a particle in a medium that is patient enough to carry it all the way to the bottom, however long that takes, without any urgency, without any destination other than the floor of the deep, which has always been there and will always be there, waiting with the quiet permanence of something that has never needed to hurry, because it has nowhere else to be, and all of geological time in which to arrive. There is a question that arises very quietly at the end of a journey like this one. Not a scientific question, exactly. Not the kind of question that can be answered with data or addressed by drilling further into the seafloor or extracting another sediment core or analyzing another set of oxygen isotope ratios from another collection of microscopic foraminifera, carefully catalogued under a laboratory microscope. Not the kind of question that belongs to any particular discipline or that sits in a journal or that has a methodology attached to it. Not the kind of question that can be approached with instruments or resolved with measurements, however precise those measurements become. It is the kind of question that arises when you have been in the presence of something very large for a very long time, and you begin to feel, not quite consciously, but somewhere beneath the level of deliberate thought, that the largeness is trying to tell you something. That the vastness of what you have been contemplating is not simply a collection of facts arranged in chronological order and narrated in a calm voice over slow music in the quiet hours of a night that is gradually becoming something other than ordinary, but something else entirely, something that has a quality, a texture, a weight that the facts themselves do not fully capture and that the chronology does not adequately convey. What does it mean to contemplate deep time, not to analyze it, not to study it or measure it or organize it into periods and epochs and events and mass extinctions and radiations and recoveries, the way a geologist organizes it in a stratigraphy column, or the way we have organized it tonight into blocks and sections and word counts, but to genuinely contemplate it, to hold in the mind for as long as the mind can manage before it retreats to the familiar scale of hours and days and human lifespans, the sheer duration of what we have been exploring. To feel, if not understand, what it means that the Ordovician Ocean was alive and teeming and complex and beautiful for tens of millions of years before the first thing that could be called a vertebrate swam through it, and that those tens of millions of years represent a span of time so much larger than anything our species has existed within that the comparison is almost meaningless. 480 million years. That is how long ago the Iapetus Ocean spread across 85% of the Earth's continental surface. Warm and amber-lit and alive with trilobites and nautiloids and graptolite colonies drifting on the current. 480 million years is a number that the human mind cannot genuinely comprehend. Not really, not in the visceral, embodied way that we comprehend things we have direct experience of. We can say the number, we can write it, we can nod at it in the calm of recognition. But to feel it, to actually feel the weight of 480 million years, the way you feel the weight of an afternoon, the way you feel the particular heaviness of a late summer evening when the light is going gold and the air is still. is beyond what the human nervous system was built to do. Because the human nervous system was built for a world measured in seasons and lifespans and generations, not geological epochs. And yet, there is something available in the attempt. In the reaching toward that comprehension, even in the full knowledge that you cannot complete the reach, that the scale you are reaching toward will remain ultimately beyond the grasp of any individual mind. Something happens in the space between the familiar scale of a human life and the incomprehensible scale of deep time. Something opens, a perspective shift that is not quite intellectual and not quite emotional, but partakes of both. A moment when the boundary between the self and the very large briefly becomes permeable, and what comes through that briefly permeable boundary is not overwhelming, but clarifying. Not crushing, but, in some way that is difficult to articulate precisely, quietly liberating. Now when you genuinely reach toward deep time, toward the image of an Ordovician nautiloid drifting through 480 million-year-old water, or toward the microbial mat pulsing in the anoxic sediment of a Cambrian abyss where no light has ever reached and no observer has ever been and no sound has ever carried except the slow percussion of chemistry proceeding through geological time. What you find is not alienation from the ordinary world you live in. You find a different kind of kinship with the world you have always inhabited, but perhaps not always seen clearly. Not the kinship of resemblance, not the kinship that comes from recognizing yourself in something, from finding familiarity in the strange. The kinship that deep time offers is older and quieter than that. It is the kinship of continuity. The recognition that the atoms that form your body tonight have been part of this story since before the story had observers. Your carbon was once dissolved in the Cretaceous Ocean. Your oxygen passed through the metabolisms of Mesozoic marine organisms. The iron in your blood carries the electrochemical heritage of ancient ocean chemistry, of the great oxygenation event 2.4 billion years ago, when the cyanobacteria of the Proterozoic ocean began producing oxygen as a metabolic byproduct in quantities large enough to first poison and then transform the atmosphere of this entire planet, setting in motion the chain of consequences that would eventually produce the oxygen-rich air that your lungs are drawing on right now, in this quiet room, in this quiet night, in this brief biological moment that is also, in its own way, are a chapter in the story we have been telling tonight. You are not separate from the ancient ocean. You are made of it. Not metaphorically, not in the loose, resonant sense of a comparison chosen for its beauty. Literally. The ocean has remade itself again and again across the hundreds of millions of years of its history. And in each of those remakings, it has produced new forms from old materials, new complexity from ancient chemistry, new observers from the lineages that survived in the deep when the surface world reset above them. The organisms of the Cambrian deep sea were made of the same basic molecular components as the organisms that preceded them in the Proterozoic ocean. The Ordovician nautiloids were made of refined and reorganized versions of those Cambrian components. The Devonian sharks carried in their cartilage and their blood the inherited chemistry of lineages stretching back to the Cambrian explosion. And the modern organisms of the deep sea, the coelacanths, the vampire squid, the nautiluses, the priapulid worms, the brittle stars carpeting the abyssal plain, carry within their cells the accumulated inheritance of every transition, every extinction, every recovery, every innovation across more than 500 million years. And so do you. You are the latest chapter of a story that has been accumulating for longer than the mind can hold. Your complexity, the extraordinary precision and delicacy of the nervous system that is even now receiving these words and processing them into images and feelings, and the gentle, welcome heaviness of the approach of sleep, is a product of the same evolutionary processes that shaped the Ordovician nautiloid, the Devonian shark, the Cretaceous coelacanth, the Triassic ichthyosaur, the Eocene basilosaurid, the Miocene sperm whale. It is not separate from those processes, it is their continuation. You are what the deep ocean eventually produced over 500 million years of patient, unhurried biological experiment, when the conditions at the surface finally allowed the lineages that had been sheltering in the deep to emerge, diversify, and become something that could sit in a warm room on a quiet night and contemplate the very ocean that made it. This is perhaps the deepest thing that the prehistoric ocean can offer us on a night like this one, in the long, quiet hours when the surface world is dark and still, and the ordinary concerns of ordinary life have receded into the soft background of a mind beginning to settle toward rest. Not just the scientific knowledge of what lived and died in its depths across hundreds of millions of years. Not just the evolutionary narrative of how life in the deep ocean served as a refuge and a reservoir through each catastrophe and each recovery. Not just the beautiful alien images of what those ancient seas looked like and felt like and moved like in their deepest parts, where the light never reaches and the pressure never relents and the time never hurries. But the recognition, quiet and difficult to hold, easy to lose in the noise of ordinary life, available again in the stillness of nights like this one, that you are continuous with all of it. That the boundary between yourself and the ancient world is not as fixed as the habits of ordinary thinking suggest. That the ocean's story is not something that happened before you, entirely separate from everything you are, sealed off behind the barrier of deep time. It is something that happened in order to produce everything you are. And in that sense, the ancient ocean is not past, it is present. It is here, in the chemistry of your body and the complexity of your mind and the particular way that your nervous system responds, even now, to the image of dark water and deep silence and the slow drift of ancient things through a world that has always been more patient and more vast and more extraordinarily old than any single moment of experience can fully contain. Which brings us, finally, you to the one question that sits at the very center of everything we have been exploring tonight. Not a question about prehistoric species or geological periods or the chemistry of ancient oceans. Not a question that any particular science can answer or any particular expedition can resolve or any particular sediment core can illuminate. A question that has no definitive answer and whose lack of a definitive answer is, perhaps, precisely the point. If every mass extinction in Earth's history has been followed by a recovery, if the deep ocean has served in each of those recoveries as the reservoir and the seed bank and the living archive from which new biological complexity emerged, and if that pattern has held, without exception, across more than 500 million years of catastrophe and renewal, then what does it mean that the story is still going? What does it mean that the archive is still being written right now in the cold and dark of the modern deep ocean, layer by millimeter layer, in the sediment that is accumulating tonight on the floor of the Pacific Abyss and the Atlantic Basin and the deep trenches of the Indian Ocean, recorded in the shells of tiny foraminifera and the molecular signatures of biological activity and the chemical traces of events happening at the surface of a world that the deep ocean has outlasted many times and will outlast again. What chapter is being written tonight? In the deep, in the dark, in the cold, in the ancient patience of an ocean that has never needed to hurry because it has always known, in whatever sense a body of water can be said to know anything, that it has all the time in the world. That is the question to carry into sleep if you choose to carry any question at all, and you do not need to. You can simply let it go. Let the whole vast story dissolve back into the darkness from which it came. Let the ancient world recede to wherever ancient worlds go when the mind finally stops reaching for them and simply rests, floating in its own deep, in the particular silence that belongs to the hours between midnight and morning, when the surface world is as still as it ever gets, and the deep below the deep is stiller still. The prehistoric ocean is still there, in a sense that is not metaphorical and not merely poetic, but genuinely, physically true. It is below the modern ocean, compressed into rock, dissolved into chemistry, preserved in the living cells of organisms that have been carrying its ancient biochemistry upward through the water column for hundreds of millions of years. And it is in the coelacanth, hovering in its submarine canyon, with the unhurried calm of something that has no need to hurry. And it is in the nautilus, rising from deep water in the night to feed in the shallows and descending again before dawn. And it is in the vampire squid, drifting through the oxygen minimum zone, with its photophores glowing softly and its ancient lineage intact. And it is in you, in this room, in this night, in the ancient chemistry of your blood and the deep evolutionary heritage that connects every living thing to the oceans of a world so old it existed before the concept of oldness had anything to measure itself against. Let that thought carry you now, if you are still awake enough to be carried. And if you are not, then it has already done its work, and the deep has received you, and the ancient world is holding you in the same patient, encompassing way that it has held everything that has ever entered it, without judgment, without urgency, without any need for the thing it holds to understand or acknowledge or reciprocate the holding. The ocean does not require your attention to continue being what it is, and what it has always been, and what it will continue to be. Long after this night is over, and the world has turned again toward morning, and whatever awaits on the other side of sleep, the ocean simply continues, as it has always continued, deep and dark and patient and extraordinarily, endlessly, timelessly alive. And whenever you are ready to explore another world, we will be here.